Hi, I'm Beechey-Gradwell and I'm a postdoc at the Resilient Agricultural Centre of Excellence at AgResearch Palmerston North. Today I'm in one of the containment glasshouses and I'm going to talk to you about my paper titled Storing Carbon in Leaf Lipid Sinks Enhance Perennial Ryegrass Carbon Capture, especially under high N and elevated CO2. The paper was published in the Journal of Experimental Botany in late 2019. The feed base for New Zealand's pastoral sector is perennial ryegrass. This plant species delivers highly digestible leafy material for animals to eat in a way which is balanced throughout New Zealand's growing seasons. However, there are strong economic and environmental reasons to want to improve perennial ryegrass nutritional quality and optimise animal performance. The high metabolisable energy, or HME gene technology, was developed by the Plant Biotechnology Group at AgResearch. Leaf lipids are an energy-dense class of compounds, but naturally occur in very low levels in pasture, around 2 to 4% of dry matter. With HME technology, two genes are added, cysteine oleosin and DGAT. Together, they cause additional lipids to accumulate in the form of encapsulated droplets. This trait was demonstrated first in Arabidopsis, then later in ryegrass, and has proven to be highly stable over time. A remarkable early discovery associated with HME expression was that it caused an increase in shoot growth and leaf CO2 uptake, or photosynthesis. This observation was the basis of the work in this paper. In the first part of the work described in this paper, multiple first-generation HME transformed lines were screened for leaf fatty acids, the major component of lipids, and transgenic protein levels. Then, a high-expressing HME line and a non-transformed control were studied in an experiment designed to comprehensively quantify major growth, biochemical and photosynthetic parameters under levels of two important plant resources, nitrogen and CO2. Growth rates were determined by harvesting entire plantlets at two time points and weighing them. HME plants had higher growth rates, especially at high CO2 and nitrogen. The HME fatty acids accumulated at the expense of leaf water-soluble carbohydrate, or sugar. Even at high CO2, HME leaves contained less sugar than the controls did at low CO2. Specific carbon assimilation traits were found to be upregulated in the HME leaves. HME leaves were thinner and had more open stomata, allowing more rapid transfer of CO2 to the fixation sites within the leaf. It's not surprising that a plant with higher lipid content should exhibit a greater rate of photosynthesis. Lipids contain more carbon than most plant compounds, and so, for a plant of a given size, synthesizing and maintaining higher lipids will necessitate that more carbon is assimilated. The intriguing observation for HME plants was that the increase in photosynthetic carbon gain clearly overcompensated for the strict carbon requirement of additional lipids, such that total plant, including root growth, increased. So what is happening inside HME leaves? Like many biological processes, photosynthesis is tightly regulated in order to maintain homeostasis. When the products of photosynthesis, sugars, are being rapidly used, photosynthesis must keep up. Conversely, when they are not being used, photosynthesis must slow down. Control of photosynthesis is dynamic, and it relies on plants sensing their internal state. It is well known that enhancing the activity of sugar-consuming carbon sinks, such as young leaves, reproductive structures, and storage organs, can remove inhibition of photosynthesis. In the paper, we speculate that accumulating leaf lipids could also diminish the signals that would usually down-regulate photosynthesis as part of carbon and energy metabolic homeostasis. Photosynthesis in HME plants is, in effect, blind to local carbon accumulation and can carry on unimpeded for longer. So why have other efforts to increase leaf lipids not increased photosynthesis and/or growth? Further, could other valuable soluble and polymeric compounds be engineered to accumulate in leaves while also removing feedback inhibition of photosynthesis? Creating an efficient carbon sink in metabolically active leaves is complex. New biochemical pathways must interfere minimally with desirable endogenous processes, and end products need to be metabolically inert or compartmentalized within cells. Wasteful, futile cycles of synthesis and hydrolysis should be avoided. In the paper, we speculate that encapsulated lipid droplets exhibit many of these properties, which may explain why the HME phenotype is unusual among metabolically engineered plants. Since this paper was published, we have been studying the translation of some of these traits to the field. I want to give a huge thanks to the Plant Biotechnology Group for their excellent science and innovation which laid the foundation for this work, and of course for their input into this paper. The next stage for the group is animal feeding trials. This will require crossing the HME trait into elite perennial ryegrass cultivars and bulking up seed material. The work is far from over.